Armando Hasurangan, biology and medicine videos. Please make sure to subscribe, join the forum and group for the latest videos. Please visit Facebook Armando Hasurangan. Please like. Here you can also ask questions, answer questions, and post some interesting things, including your hours. You can also change the quality settings to the highest one for better graphics. Now here we have an open view of the heart. We have the aorta and pulmonary artery leaving the heart, and the superior vein coming into the heart. Let's look at diastole, the time at which the heart fills with blood. But when we say diastole or systole, we usually refer to the ventricles. So here, diastole, we're looking at the filling of the ventricles with blood. So just recapping, we have the right atrium and the left atrium filling the right ventricle and the left ventricle with blood. So blood moves in. When the ventricles are filling with blood during diastole, there can be no blood coming from the arteries, such as the aorta or the pulmonary artery, so only from the atrium. This filling mechanism, as well as the ejecting of blood out of the heart, is because the heart muscles contract, they depolarize. However, muscles can only contract because of a special group of cells and fibers that coordinate the contraction process. These cells are known as pacemaker cells, and the most important is a sino-atrial node within the right atrium. These pacemaker cells maintain the conduction system of the heart, and they react automatically without any nerves or whatever attaching to them. They're automatic. But they also depolarize to send impulses. So for example, the sino-atrial node will depolarize and send impulses to the right atrium and the left atrium, causing it to contract, causing it to fill the ventricles with blood. Also, the impulse will travel to another important node known as the atrioventricular node, situated here. The impulse coming into the atrioventricular node will pause for a while to allow ventricles to be fully filled up. From the atrioventricular node, the impulse will travel essentially to the Purkinje fibers, which will then cause the ventricles to contract, to eject blood out of the heart. So it will cause the right ventricle and the left ventricle to contract to eject blood out of the heart. This process of ejecting blood out of the heart through the pulmonary artery and the aorta is known as systole, where the ventricles contract, eject blood out. So we have diastole and systole. Diastole is filling, systole is contracting. And during contraction, there can be no blood coming from the atrium to the ventricle. Before I continue, please know that diastole and systole can be for both atrium or ventricle, but they tend to be for ventricles. Diastole basically means filling, and systole means contraction. So atrium diastole means filling of blood to the atrium. Anyway, from the beginning of the video, I mentioned that pacemaker cells can only send action potentials, send impulses, when they depolarize. We can see how the pacemaker cell depolarize and send action potentials by looking at a membrane potential graph of the sino-atrial node, for example, the SA node. So here we have the membrane potential on the y-axis and time on the x-axis. We won't really look at time, we just want to understand what's happening. So here is how the sino-atrial node sends out action potentials, sends out impulses. We can name each of these phases with numbers. So 4, 0, and 3. But you might think, where is phase 1 and phase 2? Well, it doesn't occur with pacemaker cells because 1 and 2 are specifically for cardiac muscle cells, and we'll soon see later why. So for now, sino-atrial phases for action potential is 4, 0, and 3. Let's begin with phase 4. Phase 4 begins at about -60 millivolts and is where we have spontaneous depolarization. And as the name suggests, it's when the sino-atrial node spontaneously depolarizes, becomes more positive because of the slow influx of sodium and calcium ions, through the calcium ions coming through T and also L type channels. Phase 0 is depolarization, when we have rapid influx of calcium ions through L-type calcium channels, causing the membrane potential to become more positive till about 0 millivolts. This is an action potential, and this is when the sinoatrial node sends out the impulse, sends out the action potential, causing the atrial myocytes to depolarize, as well as sending the impulse to the pacemaker cells to depolarize. Then we have phase 3, and this is where we have repolarization, when the calcium ions stop moving inside, and then we have the efflux of potassium ions. The potassium ions leaving the cell, causing the cell to become more negative. So it's repolarizing. It will repolarize to about negative 60 millivolts, and then the calcium channels will close. And then the process continues. We have phase 4 again, spontaneous depolarization. As you can see, this process is continuous, and it has to be continuous to provide blood to our body. So these pacemaker cells, they work automatically with the same rhythm. However, the pacemaker cell's rate of depolarization can increase or decrease in response to some influence, problem, or condition. For example, an increased rate of firing of sympathetic nerve fibers will cause the rate of spontaneous depolarization of the sinoatrial node to reach the threshold more quickly, causing an increase in heart rate. So sympathetic activity will cause an increase in heart rate, and we see this during exercise, for example. In contrast, we have the parasympathetic nerve innervation firing that will cause the rate of spontaneous depolarization to decrease, so slowing the heart rate. And we see this when we eat, for example. So as you can see on this graph, the blue thing, the parasympathetic activity, is causing the spontaneous depolarization to be longer. Okay, now we know that when these pacemaker cells depolarize, they will produce an action potential which will cause the muscles to contract. This graph was specifically for the sinoatrial node action potential. However, the atrioventricular node, the AV node, and the Purkinje fibers are somewhat similar, just not as strong. When these pacemaker cells coordinate the contraction of cardiac muscle cells, the muscle cells themselves have to depolarize in order to contract, if that makes sense. So why don't we look at the muscle cells themselves, particularly the ventricle muscle cells, because they are the ones that eject blood out of the heart. So here we have the graph again, with the membrane potential on the Y and the time on the X. The changes in membrane potential of the ventricle muscle cells looks like this. So at rest, when nothing happens, it is at about 80 millivolts. But when it contracts, the muscle contract, they go up to about positive 20, somewhat like that, positive 20 millivolts. Now, unlike this sinoatrial node with phases 0, 3, and 4, the ventricles have phases 0, 1, 2, 3, 4. And this is because of the plateau phase, and we will talk about it soon. Now let's look at all these phases. So phase 0, we have rapid depolarization, the membrane potential becoming more positive by the rapid influx of sodium ions. This is the contraction of the ventricles. Then at phase 1, we have a partial repolarization when the membrane potential partially becomes more negative. This is when the sodium ions stop moving in and the potassium ions we have moving out. So potassium efflux. Then phase 2, we have the famous plateau phase, when the potassium ions are actually still moving out, but we also have calcium ions moving in. So it's like balancing each other out. So we have potassium moving out and calcium moving in. But after some time, when we reach phase 3, the calcium as well as the sodium ions, they will stop moving in and we will only have potassium moving out. And this is phase 3. It's called repolarization, becoming more negative. Now phase 4 is when the ventricle muscles essentially rest, and this phase is also known as the pacemaker depolarization phase. I want to explain why it's called pacemaker depolarization. Just try to think about it. But essentially, during phase 4, we have all the ions moving back to their original places. So the sodium and the calcium are moving out, and potassium moving back in where they belong, where they originally came from. And this is done through sodium potassium ATP pumps, calcium sodium exchanges, and calcium ATP pumps. So from this graph, we can see that when the ventricles depolarize here, it's when it contracts, and so we call this systole because it's when the ventricles are ejecting the blood, as shown by the height diagram on the left, the systole diagram on the left. And phase 4 is when the ventricles are filling with blood, so diastole. Now, important thing to know is the absolute refractory period, which is the whole duration of the action potential of the ventricle. The absolute refractory period is when a second action potential cannot be generated, and so we don't see a buildup of contractions, because otherwise we would have some form of heart attack. Now, knowing all this bit of information about systole, diastole, and when it occurs and what happens when it happens, let's look at an electrocardiogram. Now, you might see these at hospitals, the electrocardiograph, when patients are being monitored, which is very important, especially in intensive care. Now, the normal cardiac cycle time is about 0.8 seconds. You can correct me on this. And we can letter these phases during the cycle starting from P in the electrocardiogram. It's easy to remember, so P, Q, R, S, T. There's also a U, but that usually doesn't get brought up. The only thing kind of hard is to know what happens between these intervals. Now, this last P on the right should be after 0.8 seconds, but I drew it there to make a point. Now, you know when the heart makes a sound when it beats? Well, it occurs at the tip of R and T. And we can notice and visually see these heart sounds through a phonocardiogram. That's what it's called. Now, anyway, let's have a look at this ECG, electrocardiogram. From the P wave to the R wave, we have the atrial depolarization, or we can even say ventricle diastole, when essentially the right and left atrium contracts and fills the ventricles with blood. Then from the R wave, when it drops to the S, we have isovolemic contraction. This is when the valves between the atrium and ventricles close and the ventricles are contracting, but the valves out of the heart are not open. So the ventricles are getting ready to eject blood with force out of the heart. Then from S to the T wave, we have ventricle ejecting the blood out. So like just still with the contraction force that it had from the isovolemic contraction. From the tip of the T wave to the bottom of the T wave, this phase is known as isovolemic relaxation. So essentially the ventricles relax without being filled with blood. So both of the valves from the atrium and out to the heart are closed. I hope this is not too confusing. Essentially after the end of T, we have the ventricle diastole, when the ventricles are being filled with blood again. And from the P wave, we have the same process. We have atrial systole, atrial depolarization, where the ventricles are being filled with blood from the atrial muscles contracting. So we can see where atrial systole is. We can see where ventricle systole and diastole is. But where is atrial diastole? Well, it's actually behind this QRS wave here. And it's not that visible because it's not as prominent or not as strong as the ventricular systole. Hope that makes sense. So from this diagram, we can simplify it by specifically looking at the ventricle systole and diastole only. So from the R to the T wave, we have systole, ventricle contraction. And the others, on the other sides, we have ventricle diastole, when the ventricles are filling with blood. So again, we can even make this more simple by drawing the actual action potential of the ventricle myocyte. So if you remember how it looks like, it can start from just after the Q, it'll go up and go all the way to T. So this whole duration is when the ventricle is contracting and then slowly just about to relax. And then we can also draw the atrial myocyte contraction, and that begins in the P wave, because that's atrial systole, remember? So I hope from this diagram it kind of makes sense what is happening. It might be a bit complicated, so hopefully in the next video I'll draw a heart and draw what actually happens in the heart during each of these intervals, between the P and the R wave, etc., etc.